Good morning, ma'am.
Hello. Hello, students. I will switch off your microphones. Today, our topic is tuberculosis and leprosy. I will read the lecture. After them, I will ask you. Please listen, be attention, and listen it. Give me answers, and I will put marks. This is one part of our oral discussion. I checked your test. I'm really glad that you pass very well. But after oral discussion, you will have some written tasks. And at final, you will write test again. I will check how many knowledges you find here. Okay, now let's start of our lecture of tuberculosis. Okay, let's start. Tuberculosis. Tuberculosis is the most common infection diseases. It's infected a lot of people, and it's famous infection disease. You can read about this disease in some literature, in some novels. Next. The taxonomy of tuberculosis bacilli is next. Family is Mycobacteria, genus is Mycobacterium, and we divided the tuberculosis bacilli in 5 groups. Human tuberculosis bacilli is Mycobacterium tuberculosis. Bovine bacilli is Mycobacterium bovis. Murine bacilli is Mycobacterium microti. Avian is Mycobacterium avium, and gallbladder is Mycobacterium marinum. Who is not? Who is? Sana. Next, morphology. This is rod-shaped bacteria. This bacteria is slightly or straight curved rod-shaped. Position in the smell in pairs or in small clumps. This bacteria doesn't have flagellus and no motile, of course, and non-capsulated. This is gram-positive, but in this bacteria we don't use this technique staining, but you know this gram is our main technique staining in microbiology because we examined the sputum. Sputum is non-sterile, is liquid from non-sterile body site. They come from the lower respiratory tract and transfer via larynx, pharynx, and oral cavity. If you remember, the oral cavity consists a lot of different bacteria. And if we will stain sputum by gram, we cannot detect the Mycobacterium tuberculosis. For this one, we use special technique staining. It's called Zille-Nielsen. And we will see the red rod-shaped or pink rod-shaped bacteria on the blue background. It will this special technique will help us to detect the bacteria in different liquids from different body sites. Next, sometimes we can detect much granules. It is most characterized for young cultures. Cultivation. This bacteria grows so slowly, and we will not detect the bacteria after typical time of incubation, 24 hours. We should wait, and we should wait minimum 2 weeks. Typically, we can detect the first small colonies after 3 weeks of incubation, 21 days. And you should cultivate not less than 2 months, 4 weeks. From the first sentence, you can see the generation time 14 and 15 hours from when the first Mycobacterium tuberculosis start to for division, binary fission. If you remember, Escherichia coli is our bacteria from colon. Has the generation time is 20 minutes. Next, optimum temperature. This bacteria can grow from the 25 degrees till 40, but optimum temperature, more likely temperature, is 37. pH is neutral and less acid. Mycobacterium tuberculosis is obligate aerobe. Growth looks largely, and it means that this bacteria can grow only in presence of oxygen. No oxygen equals no growth. Next, Mycobacterium bovis in primary incubation is microaerophile. They need low concentration of oxygen and more better growth in presence of some other gases such as carbooxygen. And if we will reinoculate, this bacteria become the aerobe. What the media we will use? Selective and most common. And you will find this name in the different tests, maybe when you finish our university. But Mycobacterium tuberculosis bacilli should be cultivated on the Lowenstein-Janssen media. Other media are modification of this media, but World Health Organization choice is Lowenstein-Janssen. Of course, if you have in your laboratory, you can use Petrogenia, Dorset, consistent eggs, Tarsus, consistent blood, Loeffler media, consistent serum, and Pavlovsky media, consistent the potatoes. Colonies dry, rough, raised, irregular, wrinkled surface, creamy consistency, and we can use animals for cultivation of tuberculosis bacilli, and typically we use guinea pigs. And of course, in colonies is our form of colonies. Next. Sometimes when we confirm our diagnosis, we need express method for cultivation, and we use price method or method of microcultures. What we will do, we prepare the smears from the pathological material, typically sputum. On the several slides, we'll deflate it and sterilize it by firing or heating. The smear are first immersed in 6% of sulfuric acid solution during 5 minutes. After them, you should wash by sterile saline physiological solution. After them, you should put this slides in the liquid nutrient media, consisting 1 part of citrate blood and 3 parts of distilled water. After 7-10 days, we should color these slides by Zell-Nielsen and detect these micro colonies. On the right side of your screen, you can see the picture after price, see the bacilli after price method. And this bacilli looks like a ponytails, girls' tails. And under this is schematic picture in upper part of screen. In the under this picture, you can see the microscopic picture. And why is happened with bacteria? If the cord factor is main pathogenic factor, virulence factor of tuberculosis bacilli is present, and they connect and bind all bacilli around it. Next, this bacteria have high resistance to different solutions and external environment. You can see that by heating, this bacteria can be killed after 20 minutes. It's a lot of time. Direct sunlight kill bacteria after 2 hours. Next, in sputum, this bacteria can survive during 30 hours. Droplet nucleates during 10 days. After in air of the room during 8 minutes. And minus 20 degrees, they can be stored during 2 years. And this bacteria demonstrate high resistance to disinfectants, such as 5% of phenol, 15% of sulfuric acid, 3% of nitric acid, and other solutions, disinfectant solutions, but sensitive to iodine, kill it after 5 minutes, and ethanol, 80% of concentration, can kill the bacteria after 2 or 10 minutes. Now, you can see the lab identification of how we can prepare, how we can confirm the diagnosis. You should remember that all infection diseases should be confirmed by microbiological, virological, and some laboratory analysis. First, I want to explain the situation with pulmonary tuberculosis because it's most common and most emergency because it's very easy to spread in the environment and infect other people. First, what the specimen is sputum, because tuberculosis affect the lungs. You should take from the sputum 3 times from the patient, minimum 3 times. If you have doubtful results, you should repeat it, because from the 3 samples of sputum should be positive, minimum 2. If you have 1 positive result, you should repeat this test again. After them, you should prepare the smear staining by Zil-Nielsen, and you should check results in. You should examine the smear under the microscope. And in this table, you can see how many fields we should check. Minimum 100 fields. And you should check like a negative result if you detect less than 10 rod-shaped bacteria, pink rod-shaped bacteria in 100 watching fields. Positive 1+ if you will detect 3 or 9 bacilli in the smear when you check 100 watching fields. Next, if you detect 10 and more bacilli in the smear, you will check like a 2+. And 3+ when you detect 10 and more bacilli in smear in more watching fields. Of course, you can use fluorescent microscopy, add fluorescent antibodies, and you detect the bright rod-shaped bacteria on the black background. Look at this picture, what you can see here. Oh, nose. And how you can take the, why it doesn't work? Stop. Sorry. Uh-huh. 1 minute. I understand. Work. Look look at this video. How we can take the sputum from the patient. This outer rules. I hope you see it. You can see the sputum test help doctor understand if your sputum contains any tuberculosis germs. Be accurate when you pass tests. Don't collect saliva and don't collect the nasal phlegma. You should explain to your patients the right collection of the sputum. And we should transport the samples according to the rules of transportation of biological material. And we can store this sample in refrigerator in 4 or 8 degrees, not in freeze. Okay, but next time, sometimes maybe you cannot differentiate the sputum and saliva by microscopy when you check the concentration and consistence in the container. Next, what you should do, you should prepare the smear and stain by simple technique staining, such as methylene blue, colored in methylene blue, and you should count how many leukocytes, RBC or white blood cells, or epithelial cells you will detect in the smear. If you detect less than 15 leukocytes and more than 10 epithelial cells in the smear, you should check this result like saliva. And this is false result or false material. You should retake the sputum from the patient. If you detect more than 15 leukocytes, if you will see on the picture on the left side of the screen, you can see a lot of leukocytes and 2 epithelial cells. You can check this result as a sputum, and you can work, and you can stain this sample in the technique staining, the Nielsen staining, and we'll try to find the pink or red rod-shaped bacilli. Next, on the right side of the screen, you can see a few numbers of leukocytes and 1, 2, 3 epithelial cells. You can explain this result as a saliva, and you should recommend retake the sample again for next work. Why is so important? And this is microscopy pictures by Nielsen's technique staining. You can see blue background color because we use methylene blue for staining. We try to decolorizate by sulfuric acid and ethanol. After them, you color it. Also, first you use carbofuchsin. Next, you try to decolorate it by sulfuric acid and ethanol. And in the end, you color it by methylene blue. What's happening? Because this bacteria is resistant and surrounded by 3 layers of lipids, and you cannot remove the red color from this bacteria. And when you after them. Before methylene blue staining, your picture, your smear are decolorized, exclude tuberculosis bacilli, and you stain by methylene blue. You color background in the blue color, and check the red rod-shaped bacteria or pink rod-shaped bacteria, and you will see this one here together in pairs. Position in the smear. This is fluorescent microscope. It looks like very beautiful, very bright. When you use specific antibodies against the tuberculosis bacilli, which give a fluorescent light. When they connect, you will see this picture. If there is no tuberculosis bacilli, you cannot detect any bright, and you will see black background only. Next. Sometimes sometimes you will have a situation when your patient has positive X-ray test with some changes in the lungs. You can be sure that this person can be infected and can be dangerous for other people, can spread the Mycobacterium tuberculosis, but this patient pass the sputum, but you cannot find anything, any germs. Why is happening? Because you should remember the sputum is this is mucus. Liquid consists mucus, and sometimes when you prepare the smear, you can take the part of sputum which doesn't consist any bacteria. For best examination, you can use concentration method. When you such as like a sedimentation. After 10 minutes, our conference should be completed by program. But if it will be switched off, you should come back. Okay. You should take a sedimentation. How you will prepare the sedimentation, enrichment sedimentation of tuberculosis bacilli? You add the natrium OH and sputum. You add 4% of natrium hydroxide. Shake it during 20 minutes in 37 degrees incubation. After them, you centrifugate in 300 circulation during 20 minutes. After them, you should take the sedimentation from this tube and work with the sedimentation. Prepare the smear, inoculate on the Levenstein-Jensen, and inoculate in animals if you need. Next. Now I want to tell you about biochemical test for identification. First is Nielsen test. This Nielsen test helps us to detect Mycobacterium bovis and Mycobacterium tuberculosis. But this test helps us to detect the isoniazid resistance. Isoniazid is antibiotic which used for treatment the tuberculosis. What you will do? You should prepare two tubes. First tube consists the next reagents: 10% sodium bromide, 4% of aniline, and 96% of ethanol. You should add this solution in bacterial suspension. After incubation, you will detect, you should check the changes of color of the solution. If it will be yellow, it means this is Mycobacterium tuberculosis. If suspension doesn't change the color, it will be Mycobacteria bovis. Next test. Aryl sulfatase test. When you cultivate the bacteria, tuberculosis bacilli, after 21 days for differentiation, what is pathogenic Mycobacteria or non-pathogenic, you should add natrium hydroxide. You cultivate the bacteria on tripotation phenol diphtalene disulfate media. And if you detect pink color, change the color of colonies in pink, you will interpret this result like a atypical Mycobacteria. Neutral red test. Virulent strains able to bind neutral red in alkaline buffer solution. Avirulent strains are unable. Next, catalase peroxidase test. Catalase peroxidase test, when you use hydrogen peroxide in the tubes or on the slides, and add cultures. If you will detect bubbles in it, you can interpret this result as like a catalase positive. When you use peroxide, if the atypical bacteria or if the bacteria produce peroxidase, you will detect brown color in the medium. Now I want to demonstrate how to prepare catalase test. Here. On basis that, but this test is preparing catalase test by Staphylococcus aureus, but we should remember that this can be Mycobacterium tuberculosis. But how we can prepare and how can we interpret this result? Look at, please. Start. We add hydrogen peroxide on the slide. After that, we take separate colonies and mix it. Do you see any bubbles? Of course, no. It means catalase negative result. Next, you add hydrogen peroxide on the second slide. Mix it. What do you see? Bubbles in the draw. It means catalase negative. Oh, catalase positive, sorry. Positive, of course. We completed this test. Now, sorry, I want to demonstrate, continue to demonstrate it. I hope you understand, and this is dependent. What the result we can explain. If catalase positive and peroxidase negative, is atypical mycobacteria. If catalase negative, peroxidase positive, we will interpret it like a Mycobacterium tuberculosis. Next, amidase test. You prepare 2 tubes consisting first tube with the bacterial suspension, second tube with acetamide, benzamide, carbamide, nicotinamide, and pyrazinamide. Incubate in 37 degrees. After them, add next components: magnesium SO4, 2 parts of water, 1 milliliter of phenol, and half milliliter of hypochlorite, and boil it in 20 minutes. And check result. If it will be positive test for pathogenic bacteria, the color of suspension will be blue, and we can interpret it as it like atypical mycobacteria. Oh, 2 minutes we have only. Biochemical test for identification. Nitrate reduction test. We should interpret it. If positive, it will be Mycobacterium tuberculosis. If negative, Mycobacterium bovis. Next, allergic test, Mantoux reaction. This is most common serological test for detection the allergy against the tuberculosis. And you should know how to prepare and how to interpret it. You should detect hypersensitivity to tuberculin. Tuberculin is protein as a product of life of tuberculosis bacilli. You should inject 0.5 milliliter intradermal of the forearm here. After 72 hours, you should interpret the result, and interpret the result by checking the number of millimeters in diameter. If negative, no complete absence of any changes in the place of injection. Doubtful reaction if you will detect redness or pupil less than 4 millimeter. Positive reaction from 4 to 15 millimeters, and extra positive reaction if you will detect the diameter more than 17 millimeters. And treatment, we have 4 categories. Now our conference will be complete. 4 categories. And first category is for multi-resistant tuberculosis. First category who examined it at first time. First time is test positive. Second time, if it will be relapse or failure of treatment. Third category, if we have extrapulmonary tuberculosis. Prophylaxis by BCG, specific prevention, you know, and social prophylaxis when we have distance from the sick person. What about Mantoux test? Mantoux test, we use, if you remember, we use tuberculin. Tuberculin is protein, is product of life of tuberculosis bacilli. This test can be positive if a bacteria of tuberculosis live in our body. Sometimes can be positive result in normal condition after vaccination. If, for example, if we do Mantoux test for children 1 year old, which have only 1 year after injection of this vaccine, we have a positive result. But this is post-vaccinal allergy. And this is allergy to attenuated strain of Mycobacterium tuberculosis, bovis. It doesn't mean infection. But during the year, this is allergy should be decreased. But if you will detect some later degree of diameter of result Mantoux test, and after them you have extra positive result, you should explain like an infection, and you should examine this child that pass X-ray test, examine physical condition, pass analysis, and start the tuberculosis treatment. Here, prophylaxis. We use live vaccine. We use live strains, but the strains are weakened, attenuated. We call this vaccine BCG, bacteria of Calmette and Guérin. Calmette and Guérin are two scientists who prepared this vaccine. During 13 years, they made passage of the strains and prepare the weakened strain, which has antigenic properties but low virulence properties. We vaccinate all infants after 3 days of life, in 3 days of life after birth, in hand. I wrote about revaccination because in our country we do revaccination. In India, I couldn't find immunity infection non-sterile. It means that infection immunity can be works if the bacteria is present. Next picture. I found immunization schedule of India, and I want to demonstrate. This one. You can see this immunization schedule of India 2018, and we can see BCG at birth, maybe second or third day after birth. Medical workers will do this vaccine in India. But if we will check this vitamin A, but no revaccination in your country. If we compare your immunization schedule with our immunization schedule, specific prevention in Kazakhstan, this BCG, this is for first for till 4 from 1 to 4 days after birth. We did before it's 6 years before school, after negative Mantoux test. In Soviet countries, we did this vaccine 3 times: after birth, before school, and in 12 years old. Now, please switch on your microphones. This is case study I want to discuss with you. This is result of Mantoux test. During 5 years, this is different children, which have different results. Please switch on your microphone and tell me what is differences in two cases.
What do you have to tell?
This is for this question for Srujana, please. Only Srujana tell. What you see?
Ma'am, once repeat the question.
I repeat. What this is study. This results of Mantoux test in 2 children. Please, during 5 years. Please tell me what are differences in the results.
In the first child, in the first case, both the children are similar. in the second child, in the second case, it comparatively it is less compared to the first child. In the third case also it is similar, but in the fourth case, the second child has a drastic increase, and the first child it is reduced. In the fifth case, the second child has increased, and the first child it is reduced.
Can you explain this result? What this means for what's happened with second child?
He is being affected.
Infected, yes. This is infected. Why this result is so high in first years of life?
Because they are recently vaccinated and their body is just got the vaccine.
Okay, thank you. It is right answer.
Thank you, ma'am.
Next question. Next case study. Next. This will be for Divya. Divya, you can see microscopy slides of 3 samples of sputum. What you can say about this? How you can describe of it? What do you see on the pictures?
Ma'am, we can see the coughing of that in the first, second, and third sample.
What you will see? What you see?
Ma'am, we collect the sputum on the our slide testing, and we can see the colonies and different incubations.
No. This is microscopy. This is test material from the sick person without cultivation. You color it by Zell-Nielsen. How you can explain it? Do you have any variants? What do you see on these pictures? Look on the first sample. What you see here?
Ma'am, the difference between the saliva and sputum.
Yes, you should differentiate. And what you can say, this is sputum or saliva?
Ma'am, the first one is, ma'am, the sputum.
Why?
First sample.
Why you think
because
so? Because? Yeah.
Ma'am, how the colon means we can see the
What do you see on this picture? Describe it.
Presence of cells, maybe.
What the kind of cells you will see? You see here.
WBCs. WBC cells, white blood cells.
White blood cells, yes, but how many white blood cells you will see on this picture?
Ma'am, more than 10 cells means it's a type of
sputum.
More than 10 cells, but in a small area.
In small area, more than 15 are leukocytes, what we BC, yeah. But here not less than 15. Yes, but yes, now we can see
positive, but not a hyperpositive.
1, 2, 3 nucleus of epithelial cells.
Yes, ma'am.
You should think about saliva here because it's very doubtful result. You should ask the patient to retake the sputum. Next, okay, what the second sample? May I address this question to Abhishek more? Explain what you see here in second sample.
Ma'am, in second sample we can see many leukocyte cells. So I
Leukocyte
think
cells?
it might be sputum.
Yes,
Yes, ma'am.
okay. And what you can see on this picture, not only blue white blood cells, but what you can see here. Look attentionally, please.
Mucus.
Yes, mucus, yes, but look. Do you see
Ma'am,
any
there are some
bacteria?
red color.
Red color of, but what this means? You see,
Ma'am, rod-shaped bacteria.
what the kind of bacteria you see here? Red rod-shaped bacteria on the blue background.
Yes, ma'am.
What's
Yes,
the kind of the bacteria?
gram positive.
Ma'am, tuberculosis.
Who said?
Div.
Diva. Okay. Diva. I check that you give the right answers. Abhishek, we talk about tuberculosis. We talk about 3 samples. You should remember that by Nielsen
Yes.
on the blue background, if you will see red bacteria.
Ma'am, actually I can't hear you. There was some connection issue. Can you please? Hello.
Stop. I switch off. Okay, check. Where's our captain? Here. Sorry. Abhishek, what you want to say?
Ma'am, actually I didn't hear your voice. That's why I was saying to ask question again.
Okay. And next, Vidika, interpret the last picture, third sample. What do you see, and how you can explain this result? And your recommendation to the patient?
Ma'am, it's the mixture of saliva also. With
Yes.
the
Why you think so? Explain, please.
Ma'am, because saliva is just a liquid mixture and it doesn't make colonies as we can see in third sample.
Okay.
And ma'am, we can see WBCs in between them with dot shapes bacteria, ma'am. Yeah.
Okay, I'm agree. And what do you will recommend to patient?
What, ma'am? I can't hear you.
What you will recommend? You have this result. Can you tell to patient that he infected by tuberculosis?
Ma'am, there is WBC's number is less than 10.
You can or not?
Ma'am, cannot.
I think that you cannot say because you have 2 negative results and 1 positive. This is
Yes,
doubtful. You should recommend to pass this test again.
Again, ma'am, because they less.
Only 1 positive result.
Yes, ma'am.
Sorry. Next picture. Next case study. Next, what you see here and your conclusion. This question for Abhishek Agarwal.
Ma'am, in first sample, we can see it's saliva because number of nucleases are not more.
Okay, second sample.
Second sample, we can see it's not clear actually.
Maybe this is 2 or 3 epithelial cells and no
Yes, ma'am, it's also saliva.
Saliva, okay. And third sample?
Third sample, it's sputum.
Why?
No, ma'am, third sample is also saliva because number of cells are less.
Yes. Okay. Do you see any retrochepid bacteria?
What, ma'am?
Do you see any rod-red-shaped bacteria? Pink
No, ma'am.
bacteria? No. And what's your conclusion? First, what you will recommend to patient?
We cannot distinguish here, so it is we need to take a sputum of patient. So we need to tell him that
Yes.
sputum is needed.
Because this is incorrect material, only saliva present here, and saliva cannot demonstrate any tuberculosis, but
cannot give any test.
Yes, we should retake it. Okay, you agree.
Thank you, ma'am.
Next, what I have here. Oh, that's all. Okay, next, what I want to demonstrate. Now I will complete to demonstrate this PPT. And I should ask some students to put marks. This will be first. I ask you short question. You give me short answer. Pavitra. Where is Pavitra? Absent? Pavitra. Pavitra. Pavitra, what's the method of staining we use for detection the tuberculosis bacilli? Pavitra. Pavitra, if your network is slow, you can write in Zoom chat answer.
Ma'am, Pavitri is having internet problem.
Yes, if you problems with internet, I can repeat. Pavitra, did you hear me? Do you hear me, Pavitra?
And he is saying he cannot hear anything.
Uh-huh. Not good. Pavitra, did you take my message in Zoom? Prem, this question for you. Prem Panchar, what the method of staining we use for detection tuberculosis bacilli? The name. Students. Is it really a bad network?
Ma'am, some students are having bad network.
Aluri, maybe you can tell to us.
It's
Nitesh?
Nielsen.
Yes.
Nielsen method.
Okay, next question. What the medium of choice for cultivation? Nitesh.
Ma'am, for cultivation, we used to take sputum examples, ma'am, like
Okay,
on-spot
what the
sample and
medium,
early morning sample,
bacteriological
ma'am.
media you will use?
Ma'am, sputum smear, ma'am.
Yes, the sample you will use, like sputum sample, you can use, of course. But what the bacteriological media for cultivation? What the media of choice, agar of choice?
Ma'am, Jill Nielsen, agar ma'am.
No, Jill Nielsen is technique staining.
Agar
Asta.
ma'am, it's Lowenstein Jensen media,
Yes,
ma'am.
Lowenstein Jensen. Yes. Next question. To Darshan. Darshan.
Yes, ma'am.
Darshan, for you, question. Please tell me the biochemical test for differentiation Mycobacterium bovis and tuberculosis.
What, ma'am?
The name of biochemical test which differentiated Mycobacterium bovis from Mycobacterium tuberculosis.
Ma'am, nitrate addition test.
And
Nitrate addition test and
it was first test in biochemical test in my
Sir,
lecture.
amidase test. Amidase test.
Amazing test for detection atypical Mycobacteria. Before it.
Oh, no, ma'am. Ma'am, niacin test. Sorry.
Niacin test. Yes. Okay. Prem, explain us what the why we use Mantoux reaction. The aim or purpose of Mantoux reaction. Prem. The purpose of Mantoux reaction. Asta, help. Asta.
Ma'am, and hypersensitivity.
Yes, ma'am.
Aim of Mantoux reaction.
Ma'am, to detect allergy.
What? Asta.
Ma'am, for detecting the allergy, mainly Mantoux text is used.
Okay. Prem, please tell us what the kind of reaction after Mantoux test, kind of result after Mantoux test. Or Sana, help. Please be quickly, because after 10 minutes we have complete our test again.
Hello.
Ma'am, will start in 10 minutes, right? Ma'am, next conferencing will start in 10 minutes.
After 10 minutes we have lecture of leprosy, about leprosy.
Okay.
Hello.
Sana, please
Yes,
tell
ma'am.
us the possible results of Mantoux test.
Ma'am, it may be positive, negative, or false negative and false positive results. Positive
And?
results will be like it indicates hypersensitivity to tuberculin, and false positive may be due to infection with like mycobacteria, atypical mycobacteria, or by a false negative is due to the result may be inactive PPD preparation or improper injection technique. These all are the results of Mantoux test. Thank you.
Now I should, maybe we will complete this conference and start now a new conference with 14 minutes. Okay?
Okay, ma'am.
Come back after 5 minutes.